Modern biological databases are flooded with raw information. Every day, sequencing technologies read billions of nucleotides and amino acids, generating an immense, unorganized catalog of life. To extract any meaningful insight from this deluge, researchers must force this chaos into a highly rigid order. We have the hardware to sequence mountains of DNA and protein data, but a single string of A, C, T, and Gs is essentially a meaningless text file when viewed in complete isolation. To understand the biological function or origin of a newly sequenced gene, scientists must compare it directly against thousands of known sequences to locate matching structural patterns. This process relies on a foundational computational technique known as multiple sequence alignment, or MSA. The objective of MSA is to stack varying sequences on top of one another and arrange them so that matching characters align into exact vertical columns. When multiple organisms share identically conserved columns of biological code over millions of years of evolution, those regions indicate critical functional areas. Identifying these patterns allows researchers to map evolutionary history and deduce cellular mechanics. To execute these massive, complex alignments, researchers rely on an industry-standard computational algorithm called Clustal Omega. Developed as a specific architectural upgrade to earlier bioinformatics tools known as Clustal W and Clustal X, it was engineered with the capacity to process significantly larger datasets. Clustal Omega overcomes the traditional speed limits of computing by using an evolutionary guide tree. This mechanism allows it to rapidly dictate the order of alignment for thousands of sequences at once. This specific algorithmic architecture enables researchers to scale their work seamlessly, processing massive libraries of genetic data to uncover biological blueprints that would otherwise remain hidden. Sequencing a genome simply provides the raw alphabet. It requires a highly efficient alignment algorithm to actually arrange those individual letters into functional genetic code. Before any alignment begins, biological data must be organized into a standardized input format known as FASTA. A FASTA file is structurally simple. It contains a single header line to identify the data, immediately followed by the raw, continuous string of nucleotide or amino acid characters. To understand the challenge of scaling MSA, look at a simple pairwise alignment comparing exactly two text sequences. The algorithm shifts one sequence against the other, searching for the precise position that yields the highest number of matching characters, illuminating exact matches when columns align. For a modern processor, testing the possible variables and calculating the optimal alignment for just two sequences requires trivial computing power and happens almost instantaneously. But genomic research does not deal in pairs. A standard study requires aligning thousands or tens of thousands of sequences simultaneously. This introduces a distinct mathematical barrier, a combinatorial explosion. Adding more sequences to the simultaneous alignment exponentially multiplies the required comparison calculations. Attempting a brute force approach, where the computer tries to solve all possible sequence combinations at the exact same time, would require infinite processing time and reliably crash standard computing hardware. Because they fell directly into this mathematical trap, early bioinformatics tools routinely bottlenecked or failed entirely when researchers forced them to process the scale of modern datasets. To make bioinformatics scalable, engineers had to abandon simultaneous brute force calculation completely and instead design a strictly organized mathematical hierarchy. Clustal Omega solves the scaling problem with a specific architectural innovation. It calculates the similarity of the data pool before it attempts to perform the final alignment. First, the algorithm runs fast pairwise comparisons for every possible pair in the dataset. This numeric matrix maps the resulting scores, quantifying base similarity. Clustal Omega then translates these scores directly into a hierarchical structure known as a guide tree. Sequences yielding high similarity scores are clustered closely on adjacent branches, representing the evolutionary proximity of the actual organisms. This tree acts as a strict operational blueprint. It commands the algorithm to align highly similar clusters first, progressively moving up to merge wider, diverse branches. By enforcing this progressive, step-by-step mathematical path, Clustal Omega entirely bypasses the combinatorial explosion that crippled previous models. For the user, the practical workflow is remarkably straightforward. A researcher simply uploads their formatted FASTA file into the interface and specifies whether the input data is DNA or protein. From there, the software operates autonomously. It handles the complex mathematics, constructs the guide tree, and executes the final alignment without requiring manual intervention. The resulting speed allows Clustal Omega to cleanly process massive datasets containing thousands of sequences in a fraction of the time required by its predecessors. It achieves this velocity not by rushing the calculations, but by utilizing existing evolutionary relationships to perfectly organize its computational workload. The final output of this algorithm is a highly organized text grid. While it looks like a simple block of characters, this matrix is the starting point for concrete biological discovery. When researchers look closely at this aligned matrix, they scan for perfectly conserved columns, exact biological matches that persist across completely disparate species. In proteomics, these conserved regions provide the basis for functional prediction. By comparing unknown sequences to this matrix, researchers predict behavior and locate active cellular sites. These sequence alignments directly guide researchers in modeling the 3D structure of proteins, a prerequisite for drug design. In evolutionary biology, they extract different data. Researchers trace minor variations to map how specific genetics change over time. By analyzing these precise phylogenetic maps, scientists can pinpoint adaptive mutations, track environmental pressures, and accurately deduce common ancestors. Furthermore, when an entirely new organism is sequenced for the first time, researchers rely on these exact alignments to successfully annotate and categorize its genome. Predicting how a biological machine works, or tracking where it came from, relies entirely on the foundational accuracy of the MSA matrix. This accuracy becomes critical when the focus shifts to medical research, where sequence alignment is used directly to isolate the biological drivers of human disease. This graphic illustrates the comparative process. Researchers use Clustal Omega to align a genomic sequence extracted from a healthy individual directly against a sequence extracted from a diseased patient. By isolating a single mismatched nucleotide buried within an otherwise perfectly conserved region, scientists can pinpoint the exact biological mutation triggering the disease's onset. Locating these specific pathogenic mutations gives researchers the exact target they need to begin engineering highly precise genetic therapies. However, despite its capacity, Clustal Omega operates within strict mathematical boundaries and is not a universal solution for every dataset. The algorithm's core limitation lies in its architecture. If the inputted sequences are too divergent and lack a baseline of similarity, the software cannot calculate reliable pairwise scores, meaning a functional guide tree cannot be mathematically formed. When faced with extreme genetic divergence, researchers must pivot away from Clustal Omega and utilize specialized alternative alignment algorithms designed specifically for low similarity data. Yet for the vast majority of standard biological research, Clustal Omega remains the indispensable engine driving genomic discovery. Its specific combination of massive scalability, high speed, and evolutionary accuracy brings rigorous structure to rapidly expanding datasets. In the modern effort to decode biology and conquer disease, uncovering the hidden mechanics of life requires bringing order to the data first, one sequence at a time.